Hello everyone, my name is Preet Devgare from Amravati, Maharashtra. Today the topic that we are going to discuss is light reaction in photosynthesis. So as the name suggests, light reaction, so there has to be light so that this reaction will happen. Okay, light means in presence of light, so in daytime it will occur. And there is a dark reaction also, but there is a difference. Dark reaction does not have to be necessarily occurring in night time only. Dark reaction can also occur in daytime. So this light and dark are not associated with day and light. Specifically in case of dark reaction, it is not not associated with night. It can occur in daytime. Now why the name light and dark is there? Because in light reaction, light is used directly, but in dark reaction, light is not directly used. Instead of using the light directly, it is indirectly used, and the product of light reaction, to be specific, are used. Now, which are the products of light reaction? So first is oxygen, second is NADPH, third is ATP. Out of which, from our childhood, we have been listening to the fact that plants give oxygen. We breathe oxygen because of plants. So, today we will discuss where is that oxygen exactly coming from. As I told you, light reaction has three products: oxygen, NADPH, ATP, out of which oxygen gets liberated in air or in atmosphere. Okay, so let's forget about oxygen. Let's focus on NADPH and ATP. These are the two products formed from light energy used in dark energy for carbon fixation. But today our topic is not dark energy, so I won't elaborate what is carbon fixation. Today I will be talking about light reaction only. So as we have said, light reaction has NADPH and ATP as byproduct. So first is oxygen, it goes to atmosphere. Second is NADPH, third is ATP. Now NADPH and ATP will be used in dark cycle. Okay, fine. So now we will see where exactly light reaction takes place. So if you have learned the structure of thylakoid membrane in a chloroplast, you know this is a stroma, this is a thylakoid plate, another stroma, another bundle of thylakoid plate. This is called as grana, this is called as stromal lamellae, and you can call it granal lamellae also or grana lamellae. Okay, and the rest of the portion here there is stroma. Okay, so stroma, the lamellae associated with stroma is stromal lamellae, and the stacked portion, stacked means arranged one above the other, is the granal lamellae. Okay, so now we will see where is this light reaction takes place. So there are two sites at which light reaction takes place: stromal lamellae and grana lamellae, both. Okay, and light reaction has two kind of photophosphorylation, cyclic and non-cyclic. But before that we will understand what is photophosphorylation. Look at the word photophosphorylation. Photo means light, phosphorylation means formation of ATP. Okay, so in presence of light, when ATP is formed, we call it Photophosphorylation. Phosphorylation comes in ETS of mitochondria also, but that's a different thing. It's a whole new cycle that you will be studying in the respiration chapter. Right now I am not talking about phosphorylation in general, I am specifically talking about photophosphorylation. So in presence of light, if ATP is formed, it is photophosphorylation. I am saying that photophosphorylation is of two types, cyclic and non-cyclic. The word cyclic suggests that there must be some kind of a cycle. Okay, there must be some kind of a loop to be completed. So something might be starting from a starting point and coming there again. Okay, so it forms a cycle, it forms a loop. Yes, that is what is mean cyclic. And non-cyclic means if a molecule, here in case electron, when it starts from its starting point, it does not come back. It goes somewhere else. We will see where, but it does not come back. That's why the cycle is not completed. So that is non-cyclic. Okay. Now, there are something called quantasomes which we have learned. Quantasomes means a group of pigments which can fire its electron from ground state to excited state if they are coming in presence of light. So when the light falls on those pigments, known as photosynthetic pigments, when light falls on them, the electron in their ground state reaches to excited stage, and that state where the electron has reached on the outermost shell. So suppose there is a, I am drawing it here, suppose there is a nucleus of an atom and here is the electron. This is the ground state. So when light falls, this electron will jump here. This is the ground state, this is the excited stage. Okay, this is what happens in the photosystem. Now, quantasome, photosystem, light harvesting complex are almost similar to each other, except one cell, or not cell, one pigment of chlorophyll A, which is functionally differing from the rest of the chlorophyll A. We will see what is that. So as you have seen what is ground state and excited state, I am removing this and I'll draw another diagram of quantasome for you so that you will understand what I am talking about. So suppose there is a group of cells or pigment, not cells, pigments, which get excited and sends its ground state electron to excited stage electron, and they share the energy with each other like this to their next electron in a random manner. Here you can show it in a pyramidal manner also, like this, but there is no necessity that they are arranged in pyramid manner only, because if this exceptional chlorophyll A, which is mentioned in your NCERT also, is a specific chlorophyll A which is different than other chlorophyll A. Now when we say other chlorophyll A, so how many chlorophyll A are there? Near about 200 chlorophyll A. There are few chlorophyll B molecule and near about 50 carotenoids. Okay, so in total the quantasome is made up of approximately 300 pigments. Okay, so rest of the chlorophyll A that we are talking about are different from this particular chlorophyll A. Why? Because this chlorophyll A is known as reaction center. So it will gather the energy from rest of the pigments. Okay, it will get energy rich and then it will fire, or we can say eject its electron when it gathers too much of energy from the rest of the quantazone. Now this rest of the cell can be known as light harvesting complex, can be known as quantazome, okay, or there is one word, antenna, that also can be used, okay. And the whole structure including this chlorophyll A is known as photosystem, okay. Now remember this: quantazome, photosystem, rest of the pigments, exceptional chlorophyll A. Remember this much because this we will use to explain the light reaction further. So for now I am removing this. Let's come back to the location. As I said, cyclic and non-cyclic. So I told you there are two photostations, not photostation, wait. So cyclic can absorb 700 nanometer of light. So the wavelength of 700 nanometer photosystem is absorbed by PS1, photosystem 1, okay. And in cyclic photophosphorylation this particular wavelength is used, or we can say the exceptional chlorophyll A absorbs the light at 700 nanometer in cyclic photophosphorylation. But in non-cyclic photophosphorylation there are two photosystems involved. First one is 700 nanometer, that is photosystem 1, and another one is photosystem 2 which absorbs the light of 680 nanometer, okay. So in cyclic only 700, in non-cyclic 700 and 680 both, okay. Now we will see which photosystem is present where. Pay attention. Non-cyclic photosystem or non-cyclic photophosphorylation has PS1 and PS2, so it occurs on grana lamellae. So it occurs here, it occurs here, okay. But if we are talking about another one, that is cyclic, so the cyclic occurs on stroma lamellae. So it may occur here, it may occur here. So the lamellae which is exposed to stroma, this is stroma, and this lamellae is exposed to stroma, so that's why we are calling it stromal lamellae. There you will see cyclic photophosphorylation done by photosystem 1. Here in the granule portion you will see both PS1 also, PS2 also, causing non-cyclic photophosphorylation, okay. Okay, so now I am removing the whole diagram and I am telling you in detail what is cyclic, what is non-cyclic, okay. And after that, after explaining what is cyclic and non-cyclic, we will draw both here on the membrane of stroma lamellae and grana lamellae, okay. But first we will understand what is cyclic and non-cyclic. So for now I am removing it. Okay. So First one is cyclic, second one is non-cyclic. So as we have said, suppose there is photosystem 1 which can absorb 700 nanometer of light. Okay, when light falls on it, the excited electron, which is actually excited from its ground state because it is receiving light energy, so it will get excited. That electron ejects from the photosystem and it goes towards its electron acceptor. So if the electron is traveling from here, now look at the movement from low energy state to high energy state, so known as uphill movement. Uphill means low energy state to high energy state. It is traveling, and the electron then gets accepted by something called as FRS. What is FRS? It is ferrodoxin reducing substance. As the name suggests, ferrodoxin reducing substance. It means it reduces ferrodoxin. How? By giving it the electron that it has received. So it has accepted the electron and it will give that electron to the next compound, ferrodoxin. So now it has reduced ferrodoxin. Okay. After that, ferrodoxin will also pass on the electron to next protein known as cytochrome B6. From cytochrome B6, it will travel to cytochrome F. From cytochrome F, it will travel to plastocyanin. Okay. Now pay attention to this. Ferrodoxin is iron containing compound and plastocyanin is copper containing compound. Okay. CYAN, cyan means blue, cyanobacteria, CYAN, blue green algae. So here the reference is CYAN means it easy to remember. It's blue in color, plastocyanin. So when electron travels from B6 to F to plastocyanin, it enters again in your photosystem from plastocyanin to PS1. And we can see that there is a cycle formed. It begins from starting point, ends on the same point. That's why cyclic. Now pay attention to the energy level. When it was here, it was at its lowest energy level, the electron. But when it received the energy, it got ejected. So right now here at FRS, the electron has maximum amount of energy. But when it travels further, every step will cause loss of some amount of energy. Okay, and when it comes down to plastocyanin, the electron will have least amount of energy because there has been loss at each and every stage. Okay, so if electron does not have energy, why is it going forward? Because there is a relative increase in the affinity towards electron in these proteins. Less affinity, relatively more affinity, even more, and plastocyanin will show maximum affinity towards electron. So even if the electron is energy deficient, it will still be pulled by the next and next and next protein because they are showing affinity towards electron. Okay, and that's how electron travels. When that energy deficient electron comes here, in the photosystem. Again it receives light. When it receives light, it gets energized from ground state to excited stage, and when it goes to excited stage, it is ejected again. Okay, so this is how the cycle runs. This is known as cyclic photophosphorylation. Cycle we have seen here, but we are using the word cyclic photophosphorylation. Where is photophosphorylation? Photo is there. Where is phosphorylation? So at two stages specifically you can see phosphorylation. So first stage is this, where phosphorylation takes place. So ADP plus inorganic phosphate will form ATP. This is the first site of phosphorylation. This is the second site of phosphorylation. ADP plus inorganic phosphate becomes ATP, second side of phosphorylation. So if you conclude the reaction, you can say what is the product of the reaction. First product is first ATP, second product is second ADP. So net profit is 2 ATP molecule. Okay, keep this in mind. What is the product of cyclic photophosphorylation? We will write it here, 2 ATP molecule. Okay, this was all about cyclic. Now we will try to understand non-cyclic. So next is non-cyclic. In non-cyclic, there are two photosystems, 700 and 680 both. So first we will see photosystem 1. 700 nanometers of light absorbed by photosystem 1. Electron get excited and it gets ejected. Okay, accepted by FRS. Then given to ferredoxine. Ferredoxine reducing substance, then ferredoxine. But from now onwards, there is a difference between cyclic and non-cyclic. In cyclic, we have seen that from ferredoxine, in cyclic, from ferredoxine, it was going to B6F, okay, cytochrome B6F, and then it was going to plastocyanin. Here, from ferredoxine, it will go to FNR. Now, what is this FNR? So, it is ferredoxine NADP reductase. Ferredoxine NADP reductase. It's an enzyme. Okay, it's an enzyme which connects the upcoming H plus ion. We will see when they will come, but as the name suggests, reductase, so it will reduce the ferredoxine, it will reduce NADP. Okay, so when it receives the electron, FRS gives the electron to ferredoxine, ferredoxine gives the electron to FNR, that is ferredoxine NADP reductase, and this enzyme will send the electron to NADP. So the uncharged NADP, after receiving the electron from FNR, becomes charged. Okay, so first electron is given to NADP. Now hold here for a moment because we will try to understand 680 first. and then we will continue. So pay attention that one electron is donated to NADP, that's why there is a charge. Okay, now think of this. If the electron was ejected from PS1, it means now the PS1 has nothing. It's a vacant pigment. It can receive next electron because there is a probability or possibility of replacement of electron. One electron has gone, so there is a space for another electron, and we can call it electron deficient. So where is another electron coming from? So that's where photosystem 2 comes into picture. It will send its electron to photosystem 1. How? So on the arrival of light at 680 nanometer, it is absorbed. Then electron is at the excited state, so it is ejected after excitation, and here the electron acceptor is coenzyme Q. Q stands for quinone. Okay, QUINONE, quinone. So electron goes to coenzyme Q, then it goes to plastoquinone. If you go into the details of it, you will realize that there is plastoquinone A and plastoquinone B, but at NCERT level plastoquinone A and B is not mentioned, so I will just tell you about plastoquinone. It will receive the electron from coenzyme quinone. Then it will give the electron to cytochrome B6F. In some reference books, B6F can also be directly mentioned, and in some books there can be two different molecules shown, like B6 and separate F. So from B6 it goes to cytochrome F. From cytochrome F it again goes to plastocyanine. Highest affinity or lowest affinity? Highest affinity for electron, because electron does not have energy now. So plastocyanine is with highest affinity towards electron. So it will receive the electron, and now it will send that electron to photosystem 1. Now that photosystem 1, which was deficient before, it has received its electron now. Okay, so there is fulfillment of the vacancy of electron now. But there is a problem. 700 nanometer, photosystem 1 is satisfied with the electron, but that electron is coming from photosystem 2. So now photosystem 2 becomes electron deficient. So where is the electron coming from? It also have the possibility of replacement of electron. One electron is gone, one electron should be given to 680 now. So the problem is, who is the external electron donor? Because in this non-cyclic photophosphorylation, electron sources like 680 and 700 are the internal electron sources. But if the electron depletion is there, it's at 680, there should be another electron source, okay, which is external. So what is that external electron source? So pay attention here. It is known as oxygen evolving complex, also can be known as water splitting complex. Water splitting complex. Okay. Oxygen evolving complex. It means oxygen is evolved from here. This much is understood. Water splitting complex means there is splitting of water, cutting of water. Okay, water splits into something. Now we'll try to understand water splits into what exactly. So suppose NCERT has shown you water splitting in terms of one H2O molecule. We can try to understand with I try to understand the water splitting in terms of 1H2O, 2H2O, 4H2O sometimes. Okay, it's just that the equation later on changes. So I am telling you with the point of view of 2H2O, because that reaction is directly given in NCRT. That's why I am referring to 2H2O. Otherwise I would have used something else. So when 2H2O splits, it splits into 4 photon, it splits into 4 electron and O2. Okay, so O2, which is evolved oxygen evolving complex. Oxygen is getting evolved in the atmosphere, released in the atmosphere. Okay, so as I told you before, NADPH and ATP was the product, oxygen was not the product. See, in the beginning I told you. So this is the same oxygen that is getting evolved. Fine. Now the question is about electron and protons. So we are saying 680 is electron deficient, so it wants electron, it needs electron. So we can give this electron to 680. Okay, so let's give this electron to your photosystem. Okay, so electron will travel one by one through the photosystem and it will come here, NADP. What happens to the proton? Okay, so before answering the question what happens to proton, we have to understand something known as chemoosmosis, chemoosmotic theory. Okay, that we will study later. Right now let's focus on electron. So suppose electron is coming here. Okay, now this electron will also travel in this manner. So can you see there is a zigzag pattern? See, then it is going like this, and then it is going like this, then it is again going like this. So I told you, whenever the electron gets excited, it consumes energy and it gets excited, it liberates from its place or ejected from, then energy rich state, or if it is moving from low energy to high energy state, then it is uphill movement. So you can call this as uphill movement. But when energy reduces from protein to protein, as we have discussed before, it is reducing, this is known as downhill movement. So D I am writing. Again there is uphill movement. So uphill movement, again there is downhill movement, because again when electron comes here, it loses its energy. So here we can see that there is a Z formed. Okay, so there is Z formed, zigzag lining is there. So that's why this non-cyclic photophosphorylation is also known as Z scheme, because the way electron flows in zigzag manner. Okay, so now another electron is also coming from 680. It is going to 700 nanometer, then again it is getting ejected to FRS, and finally it is coming here. First electron was already there, now next electron is coming, it will put next negative charge. Okay, now pay attention. Your protons are also there. Now if electrons are electrons are moving continuously and protons are also getting formed, so inside the lumen of the thylakoid membrane there is concentration gradient of protons. So there will be too many H plus ions here, can be represented by plus. Okay, so some of them will go here. Now these photosystems are placed on the membrane, okay, and on the same membrane there is something called ATP synthase. ATP synthase, which is formed of two subunits F0 and F1. I will draw it later when I will show you the whole membrane. Then I will tell you how it looks. Okay, but from that ATP synthase, H plus, that is H plus ions, will travel and it will go outside the membrane. So when it goes here, the H plus will react with this electrons, okay, and now it will look like this. So when H plus reacts with this electron, we can rewrite it like this. NADP H and another H plus. Okay, now NADP H means only one proton was interacting with the negative charge. So there can be formation of NADP H, there can be formation of NADP H2 also. Okay, so NADP H is the product we were expecting. So here you can see NADP H is finally there. So oxygen has evolved. Yes, it has evolved. NADP has formed. Yes, it has formed. What about this ATP? NADP is also formed. ATP remains behind. So what about the ATP? So pay attention, in cyclic there were two ATPs formed, but in non-cyclic there is one ATP formed if two hydrogen ion, that is protons, travels through this ATP synthase. If two H plus, so suppose there are two protons traveling from F0 and F1, the F1 rotates, so there is conformational change in F1. So every time the two hydrogen or two proton molecules passes, this ATP synthase, as the name suggests, will produce ATP. So for every two protons it will give you one ATP, ADP plus inorganic phosphate, and it will give you ATP. Okay, remember this, for every two protons one ATP is produced. Okay, now imagine if there are two water molecules splitted, so there are four protons. I am saying for two protons one ATP. So for four protons, how many ATP? You will say 2 ATP, but that's not the case. Why? Wait. Here we are looking at plastoquinone. Okay, so plastoquinone is not only electron transferring protein, it also is a proton transferring molecule. We will see how it transfers proton later. Okay, and it is not that easy that if two protons are producing one ATP, four protons will produce 2 ATP, because there is hydrogen pump involved. In plastoquinone, it acts as a hydrogen pump. It will change the final result, the final outcome. So we will try to understand that. So first I will remove this and draw it on a membrane so that we can easily understand the same cycle again. On the membrane, Okay, so suppose there is a bilipid layer, and on that bilipid layer I will show photosystem 1, photosystem 2. Okay, so we have seen that photosystem 1 in non-cyclic, it is giving to FRS FD. From FRS and FD it will go to FNR. So we have to draw all the components first. So from this electron will go to FRS, then it will go to ferrodoxin, then it will go to FNR, and from FNR it will go to NADP. Okay, what happens here? From photosystem 2, electron is ejected and it will go to its electron acceptor, that is coenzyme Q. Then it will be transferred to plastoquinone. This is plastoquinone. From coenzyme 2 to plastoquinone, from plastoquinone to B6F. B6F. From B6F it will travel to cytochrome. No, wait. First it will go to cytochrome B6, then it will go to cytochrome F. Cytochrome B6, and then it will go to cytochrome F. From cytochrome F it will go to plastocyanin, and from plastocyanin the electron will go to photosystem 1. Okay, so it is the same pathway that we have seen before. It's just that we have to see the arrangement of all these proteins on the membrane. Okay, so my concern was when oxygen evolves, so we are considering 2H2O here. Okay, so we are sending 4 electrons through photosystem, but not all at once, one by one we will send. Then there is 4H plus. Oxygen has already evolved. Okay, so how the electron travels? From photosystem to coenzyme Q to plastoquinone, plastoquinone to cytochrome B6 to F to plastocyanin, from plastocyanin to photosystem, again from photosystem to FRS to FD, ferrodoxin, and like this it is traveling. So my concern was after going here, this H plus are traveling through something called I'll draw it here, ATP synthase. Okay, so when hydrogen travels, that is proton travels from F0 and this F1, ATPs are formed. Okay, so pay attention. When one electron travels from plastoquinone, one H plus comes inside the lumen. Because see, if proton is traveling from high concentration to low concentration area, so it is traveling along the gradient. Okay, this is movement of proton along the gradient from high concentration area to low concentration, but we want to maintain this gradient. We cannot afford the reversal of gradient. So if H plus ion is going outside, we know that this H plus ion will be used to form NADPH. Where is this H coming from? It is the same H, but is it necessary that whatever is the requirement of NADP, only that much amount of proton will go outside? No, more than that protons can go outside. Now if more than requirement, more than the required proton, other protons are also going outside, what about those protons? So now these protons, we will bring it back again in the gradient, because I said we have to maintain the gradient. We cannot afford to lose the photon, so this photon will be again brought back. Okay, but how much proton for the transfer of how much electron? So the number is 1:1. When one electron travels from plastoquinone, when one electron travels from plastoquinone, in the place of one electron, one proton comes inside. Okay, so that was the function of plastoquinone. It does not only act as electron transporter, it also act as proton transporter. Okay, so as I told you, if two protons passes through this, you will get 1 ATP. So ADP plus inorganic phosphate will give you 1 ATP. So now we have understood all the products of this light reaction. NADPH, yes, it has been produced. What about ATP? Oh, sorry, I have written ADP here. It should be ATP. Okay, so ATP is also there, NADP is also there, ATP is also there. So now we have achieved our products of light reaction. These are the two main products of light reaction. Okay, now these two products will be used in dark reaction for carbon fixation. Okay, okay, thank you. I hope you have understood this. Thank you.